So let's talk a little bit about enzymes and ATP. We've mentioned enzymes are proteins and we've mentioned ATP, but let's get into what they really do. We'll start with enzymes. Enzymes are proteins that speed up chemical reactions without being consumed in those chemical reactions. So that makes them a catalyst. Catalysts are things that help other things happen, but they aren't actually part of, in the end, whatever it is that happened. So you've got a wedding planner. They're going to help the whole process, but they're not actually part of the wedding. Enzymes are the same way. Just like the wedding planner can finish one wedding and go on and help in 5 more weddings, enzymes can help in a chemical reaction and then go on and help in future chemical reactions. Now, each chemical reaction has a unique enzyme that's going to help. And this gets back to this structure-function relationship. If you're going to help a particular chemical reaction, that's going to be based on your shape, and you're not going to be able to help a different chemical reaction. Enzymes, as I mentioned, can be reused. And anytime you see the ase at the end of a particular word, polymerase, synthase, that means that the particular molecule is an enzyme. So let's look at a simple example. Here we have sucrose, which is table sugar. And remember, this is a glucose and a fructose molecule bonded together. If we're going to break that apart, we're going to use an enzyme called sucrase. And sucrase is going to help cut this bond along with water, and so that we end up with these two molecules. We look at the ACE, so we know that it is an enzyme, and this sucrase, after it helps break this bond, can move on to another table sugar and another one and be reused over and over and over again. Now, a lot of times people assume enzymes only break things down, and this is because we think of stomach enzymes, but enzymes just facilitate chemical reactions. So we're going to have different enzymes that are going to help bond these two back together. So an enzyme just helps chemical reactions. It's not only something that breaks things down. So how do enzymes actually work? The main hypothesis is something called the induced fit model of enzyme function. This shows how this model works. So we've got an enzyme, and this enzyme's a protein, so it's a complex three-dimensional shape, and it's got an active site, and the active site is where the enzyme is going to bond with the substrate, whatever it is that it's reacting with. The substrate bonds with the enzyme. We then call it the enzyme substrate complex. It's bonded together. In this case, it's going to take this bond between the two parts of a single molecule and bend just a little bit to strain this bond so that it's easier to break. That bond is broken. Now the enzyme releases the two parts. Notice that we started over here with the substrate of a single molecule, and we ended up over here with a product of two separate molecules. This is the same reaction I just showed you. The enzyme has been released, and it hasn't physically changed shape. It can go back, find another sugar, and be used over and over and over again. So you have the substrate, the product. We have this temporary combination of enzyme substrate complex, which leads to formation of specific products. Another enzyme might take these two things, put them right next to each other so that they can bond together, and that would be an enzyme that's building sucrose. Enzymes can also be helped by cofactors. We mentioned vitamins when we were talking about food. Vitamins can help enzymes do their work. So they can help attract substrates to the enzyme, help stress the bonds, or put two substrates next to each other so they can more easily bond. So that's one of the important roles of vitamins. Enzymes can also be inhibited by other molecules. So an enzyme action can be blocked if a molecule were to come and sit in this active site. When the substrate bumps into this, it wouldn't be able to fit in here. And so you can stop the action of an enzyme by blocking its active site. And here are two brief videos giving you visuals on how enzymes function.